welcome to the first of our Young Scientists series. And I'm delighted to introduce Lindiwe Lamola, the first of our young scientists that we are showcasing. Lindiwe was born in the Free State in 1986, just the other day. She studied for her bachelor's degree in medical microbiology at the University of Free State before coming here to the University of Cape Town to complete her honours and master's degrees in human genetics. The university is immensely proud of her, and we're delighted to present her to you today to tell a little of her story, as well as conveying the science behind her PhD thesis on inherited cancer syndrome. Thank you so much.
Thank you for having me. So, good afternoon, everyone. Can you hear me? Good afternoon, everyone. Can you hear me now? Yes. Okay, great. So I'm Lindiwe, and I am from the Division of Human Genetics. It is a space over there, and I'll be talking to you about something that I'm very passionate about, and that is the genetic factors underlying development of cancer. So just a brief outline of my talk. I'll be telling you about my first encounter with cancer, try to explain what is it, what makes it so complex, and get into the genomics of cancer, and zoom down into my specific interest, which is in inherited cancers. So first, my first encounter with cancer happened when I was 14 years old, when my mom and my grandmother came back from the doctor. And my mom sat me down and said, "Your gran has lung cancer." And I was like, "What is that? What does that mean? Why do you look so sad? What does it mean?" And I think since that day, I've always had this curiosity trying to understand what exactly cancer is. And sadly, she did not survive her battle with cancer, but she, throughout all of that time during her battle, I learned a lot about cancer, even though I didn't really understand the science behind it. But these were words which were thrown out a lot, whether it's when she was coming back from the doctor, when we visited her from the hospital. And I think what shouted out to me was that cancer, is it death, or is it more? But it's not death. There's a lot into cancer, and that's my drive. That's why I want to understand what is cancer, why does cancer occur in certain people and not in others. So what is it? So Wikipedia nicely explains cancer, or describes cancer as a disease caused by uncontrolled division of abnormal cells in any part of the body. And in my thesis, I'll define it as cancer occurs as a result of accumulation of multiple mutations that promote clonal selection of cells that increase with increased aggressive behavior. So big words like mutation. Mutation just means any changes in your DNA, and clonal just means a copy of a specific cell. These two explanations mean the exact same things. The other one is more sciencey. The other one is just normal words that people use. So what makes cancer so complex? Why is it so hard for us to treat? So about 15 years ago, a paper was published looking at what we call the hallmarks of cancer, trying to make us understand the science behind this cancer, what actually makes it so difficult for us to just get rid of, in a sense. First of all, its ability to override normal cellular mechanisms. So normally, any cell in the body has a lifespan. And after a specific amount of divisions, a cell is supposed to just stop. It has done its job, and it's supposed to retire. But cancers do not do that. Cancers just keep growing because they can. Secondly, it has this ability to resist programmed cell death. So what that means is that programmed cell death just means a cell committing suicide, basically. So after you've done your job, or you're not feeling very well as a cell, you're supposed to self-destruct and just let other cells take over. But the cancer cell has this ability to say, even if it's under stress, it just keeps growing and growing. The other thing is that the cancer has the ability to escape or avoid immune destruction. And we all know that the immune system is there to protect us, is there to protect and serve and get rid of anything that is not, or that is harmful to our body, that is not useful in our body in any way. But cancers have this ability to just stand there and say, act normal, immune system is not going to see you, act normal, and it does. And the immune system does not see it, and then it just keeps growing. It goes elsewhere and it just keeps growing. The other thing is its ability to use its surroundings to continue to grow. So if the cell sees that it's not able to provide itself with enough growth factors or enough nutrients to keep growing, it just mimics the next cell next to it, and then it says, can we share your food, baby? Do you want to, let's just grow together. And it just keeps growing in that way. And lastly, it's its ability to allow invasion and metastasis. So what this means is that it's able to grow in your lung, and it just keeps growing and then decides, no, I want to go somewhere else. And then it just goes into your liver or somewhere else, and it just keeps growing there. So in this way, it makes it very difficult for us to track, very difficult for us to just spot and treat. Just to get into cancer genomics. So all cancers are a result of what we call changes in our genes. So changes, like I explained, it's what we call in science mutations. Just before I get into big words in genetics, I just need to explain a few terms. So DNA, so first of all, every cell in our body contains what we call the nucleus. Now the nucleus is like the powerhouse of the cell. It's like the brain of the cell. It has what we call a chromosome, and inside the chromosome we find the DNA. Now the DNA is the molecular basis of heredity. This is what we get from our parents and what we pass on to our children. And it encodes this genetic information that is responsible for development and function of me as a person. Now, within the DNA is what we call genes. Now, genes are the units. They're basically like sentences in a book. And then they're units of information. They tell the body which proteins to make. And these genes are made up of what we call nucleotides, which are building blocks of the DNA. Now, these building blocks, which are guanine, cytosine, adenine, and thymine, they're very important in a way. If you have a book, for instance, and your little sentences are your genes, so building blocks will be your alphabets. So with those alphabets, there's certain ones that always go together, which is your C always goes with your G, and your A always goes with a T. If there's in any way that your G happens to go with a T, then that is a mistake, that is a mutation, that is a change. And sometimes these changes have consequences, which is what we call mutation. So mutation is basically a change in your usual DNA sequence. And sometimes these mutations can be beneficial, sometimes they can be harmful, or they can be just neutral. It's something that happens, it's not going to harm you in any way. But they can also be either acquired or inherited. Acquired in that little changes that are happening to me right now as I stand here, but do not, that may result in something or not result in something later. But inherited in the sense that I got those from my mom or my dad. Now, as you can see in this figure here, you have your DNA strand. In your original copy, you have all your greens binding to your little purples and your reds binding to your yellows. But in this second copy, you have a mistake where you have your greens binding into your red, which is not supposed to happen. So this is where this mutation may be cancer predisposing or something like that. So now, when we talk about what is a predisposing mutation, this is the mutation that can either be inherited or acquired. It increases your susceptibility or predisposition of getting a specific disease or disorder, in this case, cancer. So just to get into cancer genomics. So cancer genomics altogether is just the study of human cancer genome. So looking at the DNA of the cancer, trying to look at all the genes, any mutations that can either be inherited or acquired throughout your life that contribute to the development or and the progression of the cancer. And as you can see on the figure here, you have your normal cell. My pointer's dead. So you have your normal cell over here, and then you have a genetic change. It could be inherited or it could be acquired, and then you have this change then causes the cell to become cancerous, and then it just keeps multiplying. As it doubles up, it ends up causing what we call the malignant tumor. While in normal cells, if there is no genetic change, the cell will divide, but as soon as there's enough of those, the other cell will die off because it has performed all its functions, it will die off. But in this case, the tumor keeps growing because it has that ability to just keep growing. So for most people, this process of acquiring mutations or having mutations that will result in cancer occur throughout life. So let's say you have a mutation when you're 12, another one when you're 15, but you only develop cancer much, much later in life. Those are okay. Well, but there are some individuals who inherit some mutations from their parents, and some these cancer predisposing mutations end up giving these individuals a much higher risk of developing cancer at a much earlier stage than the other individuals. And these would be probably at the age of 30, 40, 50, much, much earlier than the other individuals. And this is what we call hereditary cancers, and that's where my focus is. So inherited cancers altogether, they develop due to mutations that we inherit from our parents. Yes, we can blame our parents for that. But they only account for about 5% of all cancers worldwide. So you can imagine most of the other cancers, like the 95% of cancers that develop, is due to acquired mutations that happen, and you are likely to develop cancer much later in life. But these mutations that we inherit then can either be what we call recessive or dominant. Dominant just means you need one faulty copy of the gene to develop the disease, while recessive means you have to inherit two copies, one from mom and one from dad, for you to develop a particular disease. Now, there's a list of a lot of inherited cancer syndromes that have been described for over 100 years now. The most common being Lynch syndrome, which puts you at risk of developing cancers such as colorectal, endometrial, gastric, and small bowel. But these occur much, much later in life, like in your 40s or so. But there's also autosomal recessive inherited cancers, the ones that one requires two faulty copies of defective gene for you to develop these cancers. And I'd like to draw your attention to the last one here, the one with the very long name. It's called Constitutional Mismatch Repair Deficiency Syndrome, and that is what my project is on. So what is this very long name disease? So Constitutional Mismatch Repair Deficiency Syndrome is a very rare genetic cancer syndrome. Only less than 200 cases have been described worldwide to date, and it's autosomal recessive, meaning an individual requires two copies of the faulty gene inherited from both parents to develop this particular syndrome. The mutation or the change in DNA is found in something that we call mismatch repair genes. Now, mismatch repair genes play a very important role during in our cells altogether. What they do is that they correct all the mistakes, and these are all these changes in your DNA during what we call replication, DNA replication. Now, first, so I've been throwing a lot of words, but what is DNA replication? Replication is the process. So before a cell goes from one cell, during cell division, before one cell goes from one cell to producing a daughter cell or the next cell, what happens is that it has to make an exact copy of all the inherited material or the hereditary material, that is all your DNA that is found in the nucleus. So during this process, a DNA has to literally make a copy of itself, the exact copy of itself. But sometimes it happens that during this process where your alphabets are being put into place, a mistake happens. So now the role of the mismatch repair system is to come and correct those mistakes before the cell is then made. So during this process of DNA replication, what happens is that what happens is that, so at number one, you have this nice protein, one of the mismatch repair genes, saying, oh, wait, there is a mistake here, we need to take care of this. And then you'll have it calling other proteins to come and try and fix this mistake. So as it recruits other proteins, then as soon as they all come together, they identify the base that needs to be replaced and replace it. And then after that, your replication goes on correctly, and all the mistakes that were found are corrected. However, in individuals that have constitutional mismatch repair deficiency syndrome, this process does not happen. So if by any chance there's a mistake that is notified, well, it's not even notified, but if there is a mistake at all during DNA replication, nobody's there to notice that there's a mistake. So basically, they just let the cell, the daughter cell that is being made, accumulate all these mutations as it's made. So this is just to clarify that again. So you have a normal cell right there. And then what happens is that if the mutation happens, the cell has a somewhat damaged DNA. And then you have your DNA repair system comes and corrects whatever mistake was found, and you have a normal healthy cell again. However, if the cell does not get repaired, then you have more mutations occurring in that cell, in the cell, and then you have a cell that has accumulated DNA damage. So sometimes these mistakes or these mutations occur in important genes that play an important role in cell's life. And these could be your oncogenes or your tumor suppressor genes. So what they do, their job is to play as like a gatekeepers or bouncers that say, no, you cannot go through here, you are not well. And then they get rid of all the cells that are not functioning as it's supposed to, or the cells that are a little stressed. However, if there are mutations in these genes as well, nobody does that job. So all the cells that are being passed on or moved on, they have these mistakes in them. So then these cells can end up in three ways. First being senescent, which is the body just recognizes that there's something wrong with the cell. You need to stay there and stay dormant. So basically, it just stays. It doesn't grow. It doesn't replicate. It doesn't make copies of itself. It just stays there. The second one is apoptotic cell. So the cell, if it's apoptotic, it just commits suicide. It's like programmed cell death. The body gets rid of it. And the last bit is cancerous, which is sometimes some of these cells with a lot of damages end up being cancerous. So now just to continue about constitutional mismatch repair deficiency syndrome. So it was first described in 1999, and it has a very early age of onset, with an average age of range of 5 to 8 years. So these individuals end up having one particular characteristics about these individuals is that they have what we call neurofibromatosis type 1. So these will present in cafe au lait spots, and these are young children walking around with like little patches on their skin. But there are also other cancers that these individuals can develop if they are diagnosed with CMMR-D. And these can be brain cancers, hematological cancers, and what we call Lynch-related tumors. So most of these children do not survive for long, but those who do, by the age of 14, they've had about 2 or more cancers. So I'd like to introduce you to family 569. This is the family that started my project. So here above is what we call a family pedigree. So what it indicates, this is a pedigree. So what it indicates is the individual relations within a family. All squares represents male, all circles are females. And then if a square or a circle is colored in, it means the individual has had a disease or is affected, in this case has had cancer. And if there's a line through an individual, the individual has passed away. So in this family, and the story starts with 2 individuals who are not related at all, falling in love, having children, not realizing there's a family history on both sides of the family of this particular cancer. These individuals ended up having 2 children, one of which had inherited 2 faulty copies of this particular gene, being at risk at developing or being diagnosed with constitutional mismatch repair deficiency syndrome. This particular mutation that these individuals have is a South African specific mutation, and is in the gene called MLH1, which is one of the mismatch repair genes. Now, there's been a lot of guidelines because these children develop cancers much earlier than normal people. There's been guidelines throughout Europe and some from North America stating what we should do to take care of these individuals to ensure that we prevent the cancer from developing or to try and treat the cancer should it develop. So what I always like to highlight is this first one. It says for brain tumors, we have from the age of 2, these individuals should be subjected to an MRI at least once every 6 to 12 months. But what happened, and that's not the only that's for a 2-year-old, it seems quite hectic, but there's other things like the gastrointestinal endoscopy, and I think in this list now they have colonoscopy for a child from age of 3. These are very hectic kind of surveillance program that we have to put these individuals under, and what we're trying to do is try to come up with better ways of looking or surveillancing these individuals, managing these individuals to ensure that they do not develop cancer, or we can see if there is something that's going on in their body without putting something that is so expensive, that is so invasive. But also I have to highlight that our particular individual was put under this hectic surveillance program but still demised due to brain cancer at the age of 4. So the whole point why I'm doing this is that childhood cancers account for about 10% of all cases worldwide, and they have very low survival rate. And the reason why the survival rate is so low compared to adults is because we don't get to diagnose these individuals on time, or we don't diagnose them correctly. We don't treat them as we're supposed to, but that's because we don't understand these cancers very well. Very little knowledge is known about childhood cancers. So part of my study is then to say, okay, from conception, these individuals, we already have faulty genes that we inherit from our parents. But from the process where the first cell division happens all the way to when an individual is a normal offspring, there's a lot of mutations that occur in these individuals. So what is the rate of these mutations, specifically looking at individuals that have constitutional mismatch repair deficiency syndrome? And can we track these mutations to the type of cancer that they will develop? So the aim of my study was then to look at what are genetic factors underlying CMMR-D, looking at these 4 individuals, which is the nuclear family. So we used new technologies to investigate the DNA. So what we wanted to do is to look at mutations, of course. But had I sat down and tried to look at one gene at the time, it would take me forever, since there's more than 20,000 human genes that are known to date. So with the help of new technology, we are able to sequence or make copies of the individual's genome and look at it all at once, and it doesn't take that long. So what we're looking for is these mutations that are causing problems or that are affecting proteins and may end up causing this any kind of cancer. And we use what we call bioinformatics to analyze and pick out these mutations and see which ones are more likely to cause the disease. So altogether, after doing my analysis, there were about 26,000 mutations. That's a lot. But so now we had to narrow them down. Which ones are more likely to be disease-causing? Which ones are more likely to help the cancer to grow? Which ones are, So, and these genes are found in all of these mutations that we found were all in these genes. And as word cloud works, the bigger the font, the more mutations were found in that particular gene. So as you can see, there were a lot of genes, but some of them were, some of the particular genes had a lot more mutations in them than others. So now the question that I'm trying to answer right now is then what is the biological consequence of having these mutations, associated with these mutations? What role does each gene have in cancer development and progression? So there's challenges with regards to doing what I do, and particularly looking at the syndrome that I'm working on, it is very rare. So finding an individual with the same characteristics as the one that I worked on, the family that I worked on, is very difficult. But also there's diverse in presentation. You never know whether a child will develop a brain tumor or a leukemia or some kind of gastric cancer. So that makes it very difficult for us to try and develop some kind of guidelines to identify or even manage some of the individuals at risk. But moving forward, we're looking to to use the genetics to understand the development and the progression of cancer and try to contribute in improving the surveillance by identifying specific markers that we can go to the individual and say, okay, we can just test your blood and see if you have specific mutations and say, these say that you're more likely to develop a specific kind of cancer, instead of putting them through an intense surveillance program. So what have we learned from inherited cancers? Well, First, cancers are very complex. Nothing is easy. You can never just, there's no right answer. There's a lot of things that play a role for individuals to develop a specific kind of cancer. Genetics plays an important role as well. But also the use of new technologies have made it so much easier for us to try and identify specific genes that play a specific role in the development of the disease. But the struggle continues, because right now I'm sitting with a number of variants and a number of genes, and I have to still analyze and say which ones to really point to disease development or progression of the cancer. But also, I feel it is important, we need to understand the genetics to get appropriate treatment to cancer. We can't just develop a treatment without knowing what is the basis of a specific kind of cancer. And with that, I'd like to thank my supervisors, the co-investigators of the project, the colorectal cancer team, the individuals who are here, my friends and family, people who've helped with the analysis. And thank you for being here and listening to my talk. Thank you. That's very quick.
I just want to tell you that that was Lindiwe's first public lecture. So are you prepared to take questions?
I am.
And there's the pink shirt at the back, I think, and then here in the front.
Okay.
Hi.
Hi.
Very much, that was wonderful. I may have missed, but I'm not quite sure where you're getting your material from. How do you identify those parents, never mind their children?
Okay, so I work as part of what we call the colorectal cancer group up at UCT. So the way we met the family is that under this group, we have what we call colorectal cancer surveillance program. So we have a whole lot of families that are part of this program. And what they do is that every 2 years or every year, they have to come in for their colonoscopic surveillance because they are, they have to come in for their colonoscopic surveillance. So that's how the families are kept, that's how we keep in touch with specific families. How we got in contact with the specific family that I'm working on is that it was identified that the father's side of the family, well, the father developed kind of a cancer, a colon cancer, and then was then made as part of our group, our research project. And following that, then we identified that the side of the mother's side, well, the individual who's then the wife to this guy, was also in the family that is part of our surveillance program. And then from there, found out that they both have a specific genetic mutation that predisposes them to some kind of cancer. And then that's how we got to meet the children. And, well, I didn't do it, but the nurses and the doctors met the children and got to test them because they knew there was a 50-50 chance of having a child with CMMR-D. So the material, the actual material, I apologize, the material that we get, so we, if an individual is part of the research program, we either get blood or saliva to isolate DNA from, and then we test it. So for the children, we got saliva, and for the parents, we got blood. Consented for, of course.
The next question, sir. Could you say your question into the mic? Sorry, sir.
It's working.
Yes. Speak.
Can you hear me?
Yeah,
Okay. Sorry.
yeah, just have to bring it close to your mic.
Like that?
Yes,
Yeah.
lovely.
Thank you. A member of my family has neurofibromatosis. We have never been told that it's a cancer, so I was quite surprised. You mentioned type 1. What is the type that is the nasty one? Is it fatal, or is the treatment for it?
So I'm not an expert on neurofibromatosis. It's just that it happened to be part of this cancer that I'm working on, but I'm not sure about the yeah.
Okay, thank you.
You said, I think I understood you studied at Free State University.
Yes, undergrad.
And would you mind just giving us, from your childhood up, your sort of, just briefly, where you grew up,
Oh,
where
okay.
you studied and so on.
Okay, that's fair. So I grew up in a place called Qwaqwa in a small village called Hastunya. I went to school at Eddie College, where that's where I did my high school, but I did my the primary school small. What is that called again? I did my primary school in a close-by school, which was called Pedigree. And then after completing matric in, can't remember when that is, 2002. And then I had a year off in 2003. 2004, I started doing my undergrad. And I did different subjects then. Genetics was one of them, but it was not my major. I thought I want to be in medical science, and I thought medical microbiology is the way. However, when I applied for honors here, for honors at UCT, genetics came up, and I was like, maybe it's something that I could look into. And as soon as I got in, I was like, I need to work on something that really means something to me. And there was 3 projects in cancer, and I happened to be to work on one of them. And the first project I worked on in cancer was called gastrointestinal stromal tumors, where I had to identify mutations that cause this particular cancer, because depending on what mutation an individual had, you could get treatment, specific kind of treatment. And then for masters, I looked into Lynch syndrome, which is a specific kind of inherited syndrome as well, where you have a 70% chance of developing colorectal cancer, but there's other cancers like endometrial and small intestine tumors that these individuals develop. So that's what I worked on. Yeah, and now I'm doing my PhD in constitutional mismatch repair deficiency.
Thanks, Lindiwe. I'm not too sure whether my question is the same as a previous questioner.
Okay.
Is it possible to identify people who have this genetic, or who have a genetic predisposition to cancer?
If we know the family history, it only starts with the one unlucky individual who will end up at the doctor's office saying, the doctor's telling the person that you have this particular cancer. And the doctor will take family history to say, so is there anybody else in your family that has got this kind of cancer, and did it develop much earlier than it did on you, or something like that. And then from the family history, then we can determine if you have a chance of genetic or inherited kind of a cancer. And then if there is, then they will send a sample to us in the lab, which is where I'm based, and then we'll test for a particular mutation, which is very common in our population. And if we find the mutation, then we'll inform the patient and inform the rest of the family, and give them the option to be tested or to become part of some surveillance program.
In one of your slides, you appear to say that genetic mutations only account for 5% of all cancers, which seems incredibly low, but because you always get the impression that cancers come through the families. So can you just confirm that's how you understand it?
Okay, I just probably need to clarify that. So I said the cancers that are inherited, so the ones that we are sure that are from mom to dad, or from mom and dad to you, that you inherited the mutation that causes the disease, those account for 5%. But all the other cancers, the 95% of the cancers, are a result of mutations that we acquired throughout our lives. Okay. Does that make sense? Okay.
How do you acquire these mutations?
Okay. So during the
Another little question.
okay.
You spoke about cell division. Are we dividing as we speak?
As we speak.
All the time?
All the time. As we speak. All the time as we speak. So during yes, cell division is occurring rapidly all the time in our body, and it is during this process that you have replication, and during this replication then you have mistakes occurring. And when I say acquired, it may be during this process a mutation happens, but then it gets missed by the important genes to say the cell has a mutation in it. And that's because some of the mutations are harmless. Some of them may be beneficial.
So it's not an outside influence.
Well, there is environmental influence that things like UV, smoking, and all of those things that we've been hearing about. Yeah, so those do play a role as well.
Could you say something about the development of benign as opposed to malignant? What is the difference in development?
I'm not at all an expert in this, but my understanding, my very limited, it's actually should not even say, but I have no idea. But I just don't want to say because my understanding is very limited in that regard. So I'd rather not give you incorrect information.
So you're just dealing with
The malignant cancers, yes. Yep. That's it.
Now with so much knowledge about the human genome and the ability to do the gene sequencing and so on, there must surely be a strong case for potential parents, for couples who want to have a baby, to be sequenced and identify these problems, if you can.
Well, that has been put out, but it's something that we don't do in the lab. But if both these individuals, for instance, in this case, where they know for sure that they have this particular mutation in them, and it's they know that they can pass it on to their children, they probably can ask if they can test if the fetus or the potential child that they want to have does have these mutations before they actually have it. They probably can do it, but we don't do it in the lab. That's not what we work on. But I think it's something that parents will probably consider now that we know what is causing this particular cancer.
Can you tell me how long it takes to produce a genome for a human?
So for sequencing, less than a week.
And what does it cost?
I'm not very sure about the cost. I think it depends on how many genomes you run at a time, but altogether probably about 10, 15,000. It could be more. I actually am not sure.
And that time keeps coming down, doesn't it?
The price keeps coming down because it's much easier now to do that, and most labs have these machines that can sequence the human genome.
So could we all get sequenced and check with you?
I feel like if you want to. I won't be doing it though, but someone else will do it.
I'll speak quite loudly. How do you do the tests? You take the blood or the saliva, but what do you do in the laboratory?
Okay, so in the laboratory, as soon as we get the sample, being it saliva or a tissue itself or blood, what we do is that we do what it's called isolating DNA. And this is basically using specific chemicals to break down the cell walls and get out what's inside the nucleus, which is the DNA itself. And once we have removed the DNA from the rest of what we call additives and other parts of the cell, we then use that as a material that we sequence.
Under a microscope or
No, for what I do, no, we just put it in a machine and then we just, the analysis is one of those letters. It's basically me reading an A, T, G, C and seeing where the mistake is.
Thank you. Just to ask, you often hear of someone being ill, 3 to 6 months, maybe this, maybe that, and everyone's saying, why can't they find what's wrong with them? And eventually, when it's too late, they've got cancer. Why don't they just zone in on cancer?
It's probably because usually when it's not the, what do we call this, the symptoms. Sorry, the symptoms that one individual have before they get diagnosed with cancer, they're probably quite general. Like, for instance, an individual who would have colon cancer, they'll probably say, oh, it's something in my stomach. The doctor will probably likely treat what is in your stomach. Maybe they think it's probably like IBS or something even smaller, like, and give you something to help your stomach run or something, but not realizing what exactly is the cause of this like pain in your stomach. And only later, when they have selected out all the options to say, this is not it, then they look for cancer.
The question is, why don't we go straight and say it's for cancer?
Yeah, I don't know. That is a question to a doctor. I'm not entirely sure why we don't do that, but I would assume it's probably also part of expense. And cancer is such a scare. You don't want a person to come in with a stomach bug and you say it could be cancer. You know what I mean? So you have to start small, and as you work your way down, then you realize that it's cancer. I think that's the reason anyway.
Find a mutation in the child. Is there any way of preventing that child getting the cancer later?
Preventing is not very easy, but what we can do is put them under some surveillance, like watch over them, make sure that if there is some kind of small kind of lesion that's starting to grow, we get rid of it before it becomes big and cancerous. So if, for instance, if it's a stomach cancer, we can just remove whatever little lesion there is. But if it's something like your brain tumors or something, we'll probably have to subject the individual to probably like radiation or something to prevent cancer from growing.
So they can't do genetic engineering on the child.
So genetic engineering, that's very interesting. For cancer, I would say it's still at a very early stage because cancer is so complex. So genetic engineering works very well with other diseases because you find that there's only one mutation that's causing this disease, or there's only one gene responsible. But we're sitting with 700 genes that have a lot of mutations in them. So it's not very easy to just say, let's fix this one gene and then you'll be fine. So I think that's the reason why we're not there yet.
I just want to ask one question. I'm guardian to a kid whose mother died of cervical cancer when she was young, 29 years ago. Now, is there any link of genetical link to cervical cancer, or is that because of the populous virus, directly from the populous virus, or is there a genetic link as well?
As far as I know, there is genetic link. Yes, cervical cancer and breast cancer as well. So maybe.
So what would you do with a kid? Test?
So probably when they're old enough, the child would have to go through like pap smear checks and all of that to prevent the cancer from developing so just to keep it, to manage the individual not to develop cancer at all. So they just keep testing. I'm sorry?
Is it doing the test on the child because the mother might have had cervical cancer? Doesn't it both sides?
Then I'm not sure about that, but I would assume if the mother had.
Now, but there's a new, they're now inoculating young kids for the populous virus.
I've heard about that, yes.
So that would be a safety precaution.
That would be, yes. That would definitely be. Right, there's 2 more questions. Okay. Oh, the question.
I was interested in these mismatch repair proteins. Obviously, I don't understand about them very well, but is there any idea that they might be fruitful in the future as a way to cure things before they get out of hand?
Not that I know of, no. At the moment, we're still struggling to understand all the other cancers that we find are being caused by this particular mismatch repair proteins when they have mutations. But maybe in the future, but not far future, not anytime soon.
Do you have anything to counteract like throat cancer? Can you make a mutation to counteract?
I'm not understanding your question correctly.
People get throat cancer,
Mm-hmm.
then can you make a mutation in the gene or something to counteract throat cancer?
Oh, so I get. Yeah, so we're not there yet, but I'm sure someone is working on it somewhere out there. But no, not for my project. No.
Could you, if you have other questions, you can come down and chat to Lindiwe for a few more minutes.
Thank you. Thank you very much.